Today, hello, this is Ting Wu. Today, I'm going to introduce how to analyze my data. In our lab, we have collected some data, right, using fMRI. For this kind of project, we want to examine the brain activity associated with some psychological functions. For example, the reward processing, right, in human beings. But after you collect the data, so the next step is doing the data analysis. Right here, I will give you a brief guideline to how to manually analyze the data. If you don't understand why we need to do this step, right? So to analyze the data, we need to have all of this software. The first one we call it MATLAB, which is the data analyzing environment. So this is not free. You need license to use this software, but the cameras has this license, and you can, if you want to analyze the data, you can call IT to ask them whether they can install this software for you. If you are using the Mac the iMac computer, which is the one I am using in the lab, you already have the MATLAB installed, and then you can just open it. The second one is SPM, which is MATLAB toolbox for fMRI data analyzing. So this is free. We need the SPM 12, which is the newest version. To download this software, right? So you need to copy this link to your browser, and here you will see this. Click here, download form, and then you need to fill out this form briefly. First, select SPM 12, and then we need the module, which is modality, which is fMRI. So what's the operational system of your computer? So here I'm using the Mac OS. If you're using Windows or something, you can select the correct choices. And which MATLAB version will you be using? So here on this computer, I'm using MATLAB R 2018A. So here I just select R 2018A. And here you can fill out your name, your email, your institute if you want. So just download, and here you will see this link. Click this link, and you can download this zip file. That's it. And these two are for analyzing the imaging data. And you also need EEPROM and Excel to analyze the behavioral data, because for the fMRI, we want to examine the brain activity associated with some kind of psychological event, right? So to know when the event comes out, you need your behavioral data. So we assume you have all of the software ready, and then I want to introduce how to install SPM in MATLAB. To use SPM, you have to have MATLAB. So first, to open MATLAB window, all right? So just simply click this icon. If you have your MATLAB installed, simply click this icon, and let me quit this. Simply click here. You will see this pop-up window. Wait a little bit. Then you will see your MATLAB. So I can introduce the MATLAB briefly. Here have several parts. So here is your command window. So in this window, you can type in, ask the computer to do something for you, to use some command. But before using the command, you need to learn the language, right, of MATLAB. Let's try something very simple. For example, if you want to compute 1 plus 1, you just type in 1 plus 1, 1, and hit return. And the computer will tell you the answer is 2. That's simple, right? So this is command window. We can, on the right side of command window, we have the workspace. Here you will have all the variable you defined, or any variable you have got. So here, because we ask the computer to compute 1 plus 1, it automatically gives us a variable called ANS, which is the answer. So you have a new variable here called ANS. This is the name, and on its right side, which is value, F2 is 2. So if you double click here, and you will see a new window called variables. So it will give you all the details of this variable. For example, if we define a new variable called a equals to empty, right? It return. You will have a new variable called a. So if you double click a under this tab, you will see this all the information for this variable. And you can also type in some information here. For example, we need a column of values. For example, a is a vector, have several numbers, 1, 2, 3, 4, 5. You can just simply, right, define the variables here. So after you define, you can see all the information either here or here for all the values for the new variable a. That's easy, right? And on the left side of the command window, you will see everything under the current folder. It's similar as the finder, right, of your computer. So how to define the current folder? So you can define it here. Here is your current folder. So now my folder is on my desktop called sample data. If you want to change, for example, just simply change to desktop. Just select here, desktop, and then here you have a list of all of the files on your desktop. That's it. If you want to change to another folder, just for example, I want to change to sample data, I just click sample data. That's it. Right. So above those variables on those windows, you have some button you can click. So you don't need those. Right now, just only one you need is here, set path. If you want to install any toolbox in MATLAB, you need to do this. Click set path, and then add with subfolders, right? To select the folder that you want to install. For example, here I want to install SPM12, and I just select SPM12 and click open, because I already have, so I don't want to install again. So go to SPM, open, save, and close. That's it. Oh, before you install SPM, you need to check whether SPM is installed already. So how to check? You type in a simple comment called which SPM. Press return. Also here, because I already have SPM12 installed, it will give me an answer of a directory. Here is where I have the location of my SPM. So if you don't have SPM installed, after you click to hit return, you will get SPM not found. If you see this, you need to do this. So set path, add with subfolders, select the SPM12 folder. So just you need to know you need to unzip the SPM first, and then select the unzip folder, click open, save, and close. After that, check which SPM. So you will see a folder, right? Not SPM, not font. That's it. After you have SPM installed, let's open SPM to use, right? So to open SPM GUI, you just simply type in SPM fMRI and hit return. So now you have the SPM GUI. For this GUI, you have 3 windows. So the top left, which is the menu, you have several buttons you can click. Each button is for a specific function you want to use to analyze your data. I will introduce those functions later, one by one later. On the bottom left, which is another window, so after you select some function, something will maybe show up here and to ask you to enter some parameters or to make some choice, right? Or when you analyzing data, your progress, the bar will show here how many percent you have done, right? For example, from 0% to 100%, you're done. So here we got the graphic window. Some graphic information will show here. For example, if you want to visualize the image, and it will show it up here, the image of your brain. So yeah, so you have every software installed and you know how to use SPM. Let's go back to introduce the images, right? To introduce our data. So for our data, we have 2 different kinds of images. First one category we call this as a structural image. So structural image, we have several types of structural image. This kind of image is very clear. You can see the details, right, of the structure of your brain. So in our data set, we collect T1 image. This is one category of the structure image. So you have very high spatial resolution. And for T1 image, here you can see the green matter, right, which contains the body cell, the cell of the neuron body, the body of the neuron cell, sorry. And which is in gray, dark gray, is on the outer layer of your brain tissue. This is the brain, and you can see here the darker green is the gray matter. So all of the brain activity should be located on the green matter because we use the body cell to think about something. And on the inner side, you can see the color is brighter, which is the white matter. So if you learn neuroscience, you know the white matter is the fibers of the fabric of the neuron. So we use the white matter to transfer your signal. So we use the green matter to think, and then use the white matter to transfer the information. Here the gray matter is in gray, white matter is relatively white. And we have 1 T1 image per subject because because the structure doesn't change during your task, right? The brain is the brain, just the activity change. So how to measure the activity? We have another kind of images called EPI image. The EPI means planet image. You don't need to know the meaning. It's named by the sequence we use to acquire this kind of image. And those images are the functional MR image. That's what we are interested in. It records the activity associated with your task. And it records the both signal, means the blood oxygen depend signal, right? So when you are actively doing some task, the oxygen consuming level on one of the involvement region will increase. That means it light up, it activated. So this kind of this kind of image is very sensitive of your bold signal, BOLD. So compared to the structural image, the spatial resolution is low for the API image. In another word, it looks blurry, right? Why? Because for the T1 image, we want to know the detailed information. So we use 5 minutes to acquire one image. But for the API image, because our thought or our mental processes are very fast, we need to acquire data very fast to detect this kind of fast change of activity. So in our experiment, we just have 1 second to collect an image for the whole brain. That's why it is much more blurry compared to the structural image. 5 minutes, 1 second, right? So we have higher temporal resolution, but we have to satisfy our spatial resolution. So we have 1 second per image, but we collect 30 minutes for each subject. Means we have multiple images per subject, right? Each 1 second, we have 1 image. That's it. So if you open the image, and then you need to know which is the T1 image, which is the API image. And if they are from the same subject, you can see their shape, right, should be very, very similar. If you have a data, the T1 image and the API image, the shape are pretty different. You should know something must be wrong. Maybe they're not from the same subject, and then you need to contact the technician to ask to check what's wrong. Right. So after we know our image, we'll briefly introduce the data analyzing pipeline. So here we have 2 kinds of images introduced, T1 and API. This side is for we have the imaging data preparation, and then we prepare the data first, and then we do the pre-processing. So also we have .edat, which is the behavioral data from E-prime. We also need to do some behavioral data preparation, and then we combine them together, put them together, processed behavioral data and pre-processed imaging data, put them together to analyze the association between your behavior and the brain activity, which for each subject, which called the single subject level modeling. After we analyze the data from multiple subjects, we can do the second level, which is group level modeling. So here I can introduce one by one. First, we have your T1 data and API data. They are in the DICOM format, which is the raw format. I will introduce this one later. And you need to convert the data format first to our data processing. And after converted data, the format, you need to reorient an image to make them look to align, manually align these 2 kinds of images. After that, so the T1 goes to several pre-processing steps. First, to realignment, and then to go to coregistration. So coregistration, both T1 and API goes to coregistration. We coregistrate them together. And then both of them go to normalization. Normalized based on another standard template, which is the third kind of image, called template. So after that, the normalized EPI image will go to the smoothing, this step. And the smooth data will go to the first level modeling with the processed behavioral data. After we extracting the event, the onset of every event, we build the first level general linear modeling, and then we estimate the parameter of this model, and then we do the planet comparison, which is the contrast analysis, to examine the brain activity. For example, we contrast a condition to test the condition, the difference between conditions. For example, we have the condition called hit the button or miss the button, and then we can use the contrast to examine the brain activity when you are hit the button. Brain activity increase when you hit something, compare greater than when you missed the target, right? The contrast image will go to the second level analysis. Here we can have the whole brain analysis. This is more exploratory to identify brain regions associated with a specific brain function. Or you can go to the hypothesis-driven ROI, region of interest, ROI-based analysis to examine. So I'm interested in a certain brain, the activity in a certain brain regions. I know where is the region, and just want to examine this region. So this is the region of interesting analysis. And here, so I will introduce every step and how to do each step one by one later. So after that, I want to introduce our sample data, right? This data is from an adolescent study in our lab. We use a task called the monitoring incentive delay, called MID task. In this task, maybe you're more familiar with the task than me, or if you are interested in the task, you can check with Melissa or to read some literatures. So the data, the data is collected from ASRC, so which is the Advanced Science Center at City College. So for each subject, we have 2 runs of data. So for each run, the first run is the first half of the experiment, and the subject take a break after a short break, and then repeat the first one, which is the run 2, just to increase the data set, right? To increase the detection power. So for each run, we have 530 images. And so means we have 1,060 images in total, API images in total, and with 1 API images. So then I will introduce the data. So here I have a folder called sample data. I'll give you one example, right, for one subject. So here we have a folder. So if you open the sample data folder, there is a folder called raw.com, which is the raw data. In the data structure, right, this is the data structure. So when we are collecting the data, acquiring the data, first we have the localizer to localize your brain. You don't need to care about it. And then we collect the T1 image. After T1, we collect 2 runs of the task. The both one goal is the run 1. Both one goal repeated again is run 2. They are exactly the same. After each run, we simply collect a field map, which is for the correction of the image distortion. So after this one, we have some folders, and here, this is the bold one goal. We repeat it again, but this is the resting state. So the subject just lie down in the scanner. They didn't do anything, just take a break. This is the resting state. And the rest of folder are for the DTI images. Here, we don't need the EPI now. So, and then if after you download the data from the ARC server, you will see a list of folders for each subject. So here you have a folder called T1W1MPR0003, which is the T1 image you want to use for the following data analysis. This image is bias corrected. Maybe you don't know the meaning of this word. It means if without the bias correction, right, the outer layer of the brain will looks brighter than the subcortical regions, and the machine, the scanner, automatically applies a correction look to improve the homogeneity of your image. So the folder above the name 002 is the raw data without bias corrected, but we don't need this one. With what we need is 003. So these two folders, both one go 004 and both one go 009. They're for the task images for ROM 1 and ROM 2, respectively. And those four folders, right, are for the field map image. AP is from anterior to posterior. This is the direction of the scanning. PA is posterior to anterior. Doesn't matter. So, and for each pair of folder, the first one is the magnitude image, and the second one is the phase image. Also, the magnitude and phase for the PA direction, and these two are for the ROM 2, right? These four folders are for the ROM 2 field map images. So we may need those field map images for correcting the distortion of your image. Depends on your data quality. We may need those, or we may not need them. Okay, so now you know our data, right, and our basic pipeline. So next time I will start to introduce how to prepare your data. Right, see you next time.